Hello Keshav. Today we are going to learn about the process of replication. Before we go into the process of replication, I would like you to know something about central dogma of biology. Then we will know about something about the in which stage of cell cycle does this DNA replication process occurs. Then we will move on to the which mode of replication is used to replicate DNA, then who proved it experimentally, then moving on to the DNA machinery, like machinery and the enzymes involved in this process of replication, and basically the steps as well. So let's start with it. I will just share the screen. I hope it's visible. Okay, first I told you about the first it was what is central dogma of biology. Consider this is DNA. So there is the genetic flow of information from this DNA to RNA, and further from RNA, this genetic information goes on to the proteins. Okay. So what happens here is this first in a cell, the DNA duplicates. So here we have the process of replication. Which process? We have the process of replication. Then this cell goes into other cells and perform the process of transcription. This transcription DNA gets converted into RNA. Then from RNA, these are translated to proteins. So this is basically the central dogma of biology. Further moving in this direction, I told you that we will study about the stages of cell cycle in which this DNA replication occurs. So as we know, there are few cell cycle stages, namely G1 phase, we have S phase, we have G2 phase, and then we have the M phase. So basically DNA replication, this part is DNA replication occurs in the S phase of the cell cycle. It occurs in the S phase of the cell cycle. So basically what is the DNA replication? In simple terms, copying. Copying of what? Copying of genetic material. Copying of genetic material. What is the genetic material here? It's the DNA. And we know, as we are aware, there is the double helical model of DNA. Double helical model of DNA given by Watson and Crick in 1953. So this double helical model of DNA states that two strands of DNA are complementary, act as a template for own partners. Like consider कि मतलब दो strands हैं, two strands are there. These both strands separate. One goes this side, one goes that side, and it generate. These are template strand, and these new synthesized strands are complementary strands to it. So what we done is we duplicated DNA. You can see this is one molecule of DNA, this is another molecule of DNA, and the DNA is duplicated, and the complementary, these two were complementary to each other. They separated and both became the template strand. One template strand, second template strand, and we get a newly synthesized DNA 1, and this is newly synthesized DNA strand 2. So this was the basic definition to it. Now we will move on to the what was the mode of replication. Mode of replication was semi-conservative mode of replication. What was the mode of replication? Semi-conservative mode of replication. What is basically the semi-conservative mode of replication? Let's see. And this semi-conservative mode of replication was proved experimentally by Messelson and Stahl. Who proved it experimentally? Messelson and Stahl proved it this experiment in 1958. So this experiment is also known as Messelson and Stahl experiment. So basically when we are asked about the semi-conservative mode of replication, we need to explain this experiment of Messelson and Stahl. So let's have a look. What they did basically in this experiment was they took E. coli. They took E. coli cells and grown in ammonium chloride. Grown in ammonium chloride. So the catch here is it was heavy isotope of nitrogen. This 15, you can see here is atomic weight. Basically nitrogen's atomic weight is 14, but this is 15. So E. coli cells were grown in heavy isotope of nitrogen. What was done was heavy isotope of nitrogen. So what basically occurred was this was one DNA molecule, and this was the complementary strand to it. So both for few generations it was grown in this ammonium chloride. And they also what they did was then they centrifuged it in against the density gradient of cesium chloride. Like centrifugation against cesium chloride. I will draw these steps. You can I need you to pay attention what I draw. See this, I am drawing this one template strand was done. This was the other template strand. Okay, so this is 15 nitrogen DNA. Same here it is 15 nitrogen DNA. Okay, I am drawing it till second generation. So you can see again these two DNA strands will separate and form another DNA. So this is one. This is 2. 15. We have to mention this all. You can see this heavy isotope of nitrogen. I have mentioned that this strand was till second generation. It will be until many generations. It will be continuously transferred, but it will be only parental. So now what happened was like again this was centrifuge under the density gradient, and in the first generation we got the heavy band of nitrogen. So this was the heavy band. Let's complete this diagram of the experimental diagram. Okay. Okay, see After 20 minutes, what happened was this two DNA was separated and we had a complementary strand to it. This was 14 nitrogen DNA. How it occurred was this E. coli cells. What happened was this E. coli cells were transferred from ammonium chloride, a heavy isotope of ammonium chloride, to simple nitrogen ammonium chloride. It was transferred to 14N H4Cl ammonium chloride medium. E. coli cells were transferred to it. So we will get this is after 20 minutes, it was like generation 1, and after 40 minutes, it was generation 2. As we know that E. coli cells take 20 minutes to reproduce. So this was generation 2. Simply here also the E. coli cells were placed in only simple ammonium chloride. So this was second generation strand with simple nitrogen, and what we got here was a hybrid band. This was, you can see, this is a hybrid band, and in the generation 2, you can see this is you. I hope you are paying attention to the diagram, whatever I am drawing step by step. You see this is 14 and nitrogen, and 2 more strands were synthesized. You see these both strands are 14 and DNA, 14 and DNA. Okay, similarly here 14 and DNA. What was the conclusion here? That we got a hybrid band here and one light band this side in the density gradient centrifugation. So let's see this here. Okay, let's have a quick revision. So what happened here was E. coli cells were grown in ammonium chloride of heavy isotope of nitrogen for many generations. So we got both the strands of this nitrogen got incorporated into the nucleic acid, and it was a heavy isotope. It's 15 and DNA. Both strands were covered in it. Then we performed the density gradient centrifugation, and we got a heavy band. What we got was heavy band. Then after 20 minutes, generation 1 was obtained, and we got like these cells, E. coli cells were transferred from culture medium of heavy isotope to simple ammonium chloride, simple nitrogen ammonium chloride medium. And after one generation, we obtained like 20 minutes after we obtained this, one strand was template strand, another was the complementary. And after we done the density gradient centrifugation, we obtained the hybrid band. Similar, and in then the medium was on change. It was only in the simple ammonium chloride medium. Then again we got 4 DNA molecules, one with template strands, each one with the template strand, one with the complementary strand. You can see 1 2, these 1 2 are the 15N DNA and rest are 14N DNA. Again the density gradient centrifugation against cesium chloride was obtained, and it was found that this time we obtained was one was hybrid and one was light. Had it been conservative mode of replication, so we would get a light band and heavy band, but since we got a light band and hybrid band, so it is known as semi-conservative mode of replication. Okay, this was it for the Michelson and Stall experiment. We now know that this is DNA replication occurs by semi-conservative mode of replication. Moving on to further, now we need to know how this DNA replication process occurs. So consider what are the enzymes involved in. First, we will look into the enzymes. First enzyme is helicase. What is the process, like what is the function of this helicase? It unwinds the DNA. What it does is unwinds the DNA. Second is topoisomerase. Topoisomerase. What topoisomerase does is it cuts and seals DNA. I'm just basically mentioning here one by one, then we will see step by step procedure. This is helicase, then topoisomerase cuts and seals DNA. Then we have primase. What it does is it synthesizes primers. Primers synthesis. What are these primers? These are single-stranded DNA molecules which helps in polymerization further. So fourth is DNA dependent. Fourth is DNA dependent DNA polymerase. DNA dependent DNA polymerase. What it does is it polymerizes DNA in 5' to 3' direction. What it does is synthesizes 5 in new strand in 5' to 3' direction DNA synthesis. This is fourth one. Then we have another enzyme, DNA ligase. DNA ligase. This is known as molecular glue. So what it does is it ligates DNA when it's ligates DNA when it is synthesized discontinuously. So these are the enzymes, and another one it's not an enzyme but it's a substrate here. It's a DNTP. So what is this DNTP? Deoxyribonucleoside, ribonucleoside triphosphate. Deoxyribonucleoside triphosphate. So basically what it does is it acts as substrate. It will be added to the 3-OH site and it will help to polymerize DNA. This is substrate, and also it gives the energy in the form of ATP. Okay, so these are the enzymes and the machinery involved in DNA replication process. One more thing, like if consider this is a DNA strand, 3' to 5' and 5' to 3'. So what basically here is we have the ori site. Here we have the ori site. What is ori site? Since this is an E. coli genome, so we will say that there are definite regions in the E. coli genome from where initiation of replication occurs. Initiation of replication occurs. Initiation of replication occurs from this site. So let's see this site. Okay, Consider now we will beginning with the steps of the process of replication. Consider this is like this part you see. Now rotate anti-clockwise. Okay, anti-clockwise 90 degree. If we rotate it just for the perspective, we will see what happens. See, so this was this DNA strand. As we know, like what now you have to give the directionality. What like 3 dash to where is the 3 dash to 5 dash? As we know that like 5 dash to 3 dash it's synthesis now. So this will be 3 dash 5 dash 3 dash 5 dash. What happens is this. Now consider key here you see. Now consider helicase occurs here. Helicase comes here. What comes? Helicase comes. Helicase comes and binds here, and what happens? This Y-shaped replication fork is formed. What is formed is this is Y-shaped replication fork. This is Y-shaped replication fork. Again, don't forget to mention the polarity. This is 3 dash, this is 5 dash, 3 dash to 5 dash. Okay, then topoisomerase. What happens is topoisomerase. What does is topoisomerase seals the DNA here, seals the DNA so that no further from the ORI side doesn't replicate till it's still the replication occurs till here itself. Okay, so now comes the primase. Okay. Here now consider that the primase has synthesized the primers. What are the primers? They are single stranded. One second, just a second. We see that here we got the primers, and here also we will get the primers. Primer. Now these primers will synthesize the DNA. Now comes the DNA dependent DNA polymerase. So DNA dependent DNA polymerase synthesizes DNA in they synthesize DNA in 5 dash to 3 dash direction. Since this is 5 dash direction, so it synthesizes DNA in 5 dash to 3 dash direction. This is 3 dash direction, and the synthesis occurs continuously. This is known as continuous synthesis, and this is the leading strand. So the DNA is synthesized continuously in leading strand. This is a leading strand where continuous synthesis occurs. Now you see this is the primer 5 dash. See here it was 3 dash, so now it will be here 5 dash to 3 dash synthesis occurred. Now this is again 5 dash here, further synthesis occurs till 3 dash. Now again 5 dash till here. What is 3 dash? So you can see there is discontinuous synthesis. What occurs is discontinuous synthesis. Discontinuous synthesis, and this strand is known as lagging strand. DNA polymerizes discontinuously in this lagging strand. So you can see these three fragments are formed. These are known as Okazaki fragments. What they are known as? Okazaki fragments. These Okazaki fragments are ligated. Okay, these are what happens. These are ligated. These are ligated by okay, just a second. Okay, these are ligated by DNA ligase. Okay, DNA ligase occurs to ligate these Okazaki fragments, and also that's why it is known as the molecular glue. And these primers also were removed by DNA polymerase. After these, everything has been ligated and polymerized. What comes here is topoisomerase. Topoisomerase cuts the DNA and we get a whole duplicated DNA. Also, then DNA polymerase comes to occur for proofreading. What it does is DNA polymerase does is proofreading. So we can add functions, few of the functions here, like DNA synthesis and also proofreading. Proofreading, there is proofreading activity, there is 3' to 5' exonuclease activity. Actually, this is what it does is it happens to remove those primers too. Then again, it DNA ligates with it. So let's have a quick revision. What we followed till here was we seen central dogma of biology. Then when we move to the DNA replication definition, we happen to know about the semi-conservative mode of replication and the Michelson and Stall experiment. Okay, in what it was concluded semi-conservative the mode of replication. Then we came to know about the enzymes involved and machinery involved in DNA replication. First was the helicase enzyme. Helicase, what was known as? It unwinds the DNA. Topoisomerase was known to cut and seal DNA. Primase was known to synthesize primers. Then we had the DNA dependent DNA polymerase. So it basically add on to those primers. The substrate was DNTP, deoxyribonucleoside triphosphate, and then we had the DNA ligase, molecular glue. Here we seen the like DNA strand where we got the ori site. Ori site is origin of replication. What is ori full form? Origin of replication. So it's basically origin of replication site, ori site. We made an anti-clockwise 90-degree rotation to better understand it. See, now polarity we gave 3' to 5' with 5' to 3' after making it anti-clockwise. So then came the helicase enzyme came, and what it did was it basically unzipped the DNA. Like means you have seen a chain, it unzips the DNA, making it an Y-shaped structure. So it's known as Y-shaped replication fork. As you can see, like it's an inverted fork or it can be fork. So it's a Y-shaped fork, inverted Y, and directionality or polarity we can say 3' to 5'. Here 3' to 5'. Then it came, what it came was topoisomerase to seal the DNA here. Then came the DNA like the primase from these primers here, and then came the DNA polymerase, DNA dependent DNA polymerase. So here was DNA and DNA dependent DNA polymerase. It continuously synthesized the DNA to 5' to 3' direction. So it's known as continuous synthesis in a leading strand, and it the DNA was synthesized discontinuously. This side you can see 1, 2, 3 in 5' to 3' direction only. So you can see that polarity is also there, 3' to 5'. Whenever you make in exams, don't happen to make this mistake. So you better know that key it happens to be in 5' to 3' direction. So here if it's continuous 5' to 3', here will be 3'. Then you can place 5' here, 3' here, and then 5' here. So, and then here it's 3' direction, so it will be 5' here. So here it is DNA dependent DNA polymerase synthesizes discontinuously in the lagging strand, producing Okazaki fragments. These Okazaki fragments are ligated by DNA ligase known as molecular glue, and then topoisomerase cuts this DNA, and the DNA ligase and the DNA polymerase, DNA dependent DNA polymerase also perform the function of proofreading. So it proofreads the DNA, it's whether it's synthesized properly or not. So that's all for this process of replication. I will stop sharing the screen. Yes. Any questions are most welcome here. So what was the DNA replication process? It was like, as we seen, duplicating the DNA, generating a complementary strand from the template strand. Then we seen the Michelson and Stahl experiment, in which it was the equalized cells were transferred from ammonium chloride heavy isotope to simple nitrogen. First they were generated for generations, for many generations they were done in ammonium chloride of heavy isotope. Then it was transferred to simple ammonium chloride. So in that, then generation 1 we got generation 2, and we basically made a density gradient centrifugation. So it was it was basically done to separation of DNA by density gradient against the cesium chloride. So we got the heavy band, we got, then generation 1 replication we got hybrid band, and then generation 2 replication we got a hybrid and the light band. So you basically see, had it been conservative, so we wouldn't get the light and hybrid band, we would got a light and heavy band. Instead we got light and hybrid band. So it proves that the DNA was replicated semi-conservatively with one parental strand and one the newly synthesized strand. So after that we go seen the enzymes involved and the DNA machinery involved in the process of replication. All the enzymes are helicase, topoisomerase, DNA dependent DNA polymerase, primase, ligase, and the substrate was deoxyribonucleoside triphosphate, DNTP is what we say. It basically acts as the substrate and also provides the energy for this like process of replication. Then we go on to the steps of replication. Uh, it's like first the helicase comes, binds to the DNA, it unwinds and forms a Y-shaped structure, like unzipping of DNA, and a Y-shaped structure is formed, inverted Y. Then topoisomerase seals, like this shape has to be sealed na here कि like इसके आगे नहीं. So it seals the DNA. Then primase comes and makes the primers so that the DNA is synthesized easily. Then those primers are polymerized with the DNA. Substrate DNTPs comes and DNA dependent DNA polymerase continuously synthesized in the lagging strand and discontinuously synthesized in lagging strand discontinuously. Those fragments are joined, Okazaki fragments are joined by DNA ligase. Then the topoisomerase cuts the DNA, and at the last, DNA dependent DNA polymerase proofreads the DNA that it is replicated properly or not. Otherwise, we have to terminate, like this cell has to die, otherwise it may cause mutations in the cell. So hope you have understood this process of replication well. All the best. Thank you.